Hello, welcome to Biology, the scientific study of life. Again, more about cytoskeleton. Let's complete cytoskeleton and the components of the cytoskeleton. There are three components: microtubules, another name also called tubulin polymers, and microfilaments, also actin filaments, the same. Intermediate filaments, these are the component of the cytoskeleton. Okay, let's talk about filaments or microfilaments, actin filaments. Microfilaments are solid rods about 7 nanometer in diameter. They are also called actin filaments because they are built from molecules of actin. A globular protein, a microfilament, is a twisted double chain of actin subunits. Besides occurring as linear filaments, microfilaments can form structural networks due to the presence of proteins that bind along the side of an actin filament and allow a new filament to extend as a branch. Microfilaments seem to be present in all eukaryotic cells. In contrast to the compression-resisting rule of microtubules, the structural rule of microfilaments in the cytoskeleton is to bear tension, which means pulling forces. The ability of microfilaments to form a three-dimensional network just inside the plasma membrane helps support the cell's shape. This network gives the cortex, which means outer cytoplasmic layer of a cell, the semi-solid consistency of a gel, in contrast with the more fluid, which means solid state, of the anterior cytoplasm in animal cells specialized for transporting materials across the plasma membrane, such as intestinal cells. Bundles of microfilaments make up the core of microvilli, the previously mentioned delicate projections that increase the cell surface area. Microfilaments are well known for their role in cell motility, particularly as part of the contractile apparatus of muscle. Notice that muscle cells, thousands of actin filaments are arranged parallel to one another along the length of a muscle cells. Notice that muscle cells interdigitated with thicker filaments made of a protein called myosin. Myosin acts as a motor protein by means of projections arms that walk along the actin filaments. Notice that. Contraction of the muscle cells results from the actin and myosin filaments sliding past one another in this way, shortening the cell. On other kinds of cells, actin filaments are associated with myosin in miniature and less elaborate versions of the arrangement in muscle cells. These actin myosin aggregates are responsible for localized contractions of cells. For example, a contracting bolt microfilaments forms a cleavage furrow that pinches a dividing animal cell into two daughter cells. Localized contraction brought about by actin and myosin also plays a role in amoeboid movement. Notice that in which a cell such as an amoeba, for example, crawls along a surface by extending and following into cellular extensions called pseudopodia, from the Greek pseudos, false, which means, and bod, which means foot. Pseudopodia extend and contract through the reversible assembly of actin subunits into microfilaments and of microfilaments into networks that convert cytoplasm from cell to gel. According to a widely accepted model, filaments near the cells trailing and interact with myosin, causing contraction like sequencing on a toothpaste tube. This contraction forces the interior fluid into the pseudopodium, where the actin network has been weakened. The pseudopodium extends until the actin resembles into a network. Amoeba are not the only cells that move by crawling, so do many cells in the animal body, including some white blood cells. In a plant cell, both actin-myosin interactions and sol-gel transformations brought about by actin may be involved in cytoplasmic streaming, circular flow of cytoplasm within cells. This movement, which is especially common in large plant cells, speeds the distribution of materials within the cell. Intermediate filaments, the third component of cytoskeleton. Intermediate filaments are named for their diameter, which at 8 to 12 nanometer is larger than the diameter of microfilaments, but smaller than that of microtubules. Notice that. Back to the diagram. This one, intermediate filaments, notice the size bigger than microfilaments and smaller than microtubules. Back to intermediate filaments, also specialized for bearing tension, like microfilaments. Intermediate filaments are a diverse class of cytoskeletal elements. Each type is constructed from a different molecular subunits belonging to a family of proteins whose members include the keratins. Microtubules and microfilaments, in contrast, are consistent in diameter and composition in all eukaryotic cells. Intermediate filaments are more prominent fixture of cells than are microfilaments and microtubules, which are often disassembled and reassembled in various parts of the cell. Even after cell die, intermediate filaments network often persists. For example, the outer layer of our skin consists of dead skin cells full of keratin proteins. Chemical treatments that remove microfilaments and microtubules from the cytoplasm of living cells leave a web of intermediate filaments that retains its original shape. Such experiments suggest that intermediate filaments are especially important in reinforcing the shape of a cell, fixing the position of certain organs. For example, the nucleus commonly sits within a cage made of intermediate filaments, fixed in a location by branches of the filaments that extend into cytoplasm. Other intermediate filaments make up the nuclear lumina that lines the interior of the nuclear envelope. In cases where the shape of the entire cell is correlated with function, intermediate filaments support the shape. For instance, the long extensions axons, which means of nerve cells that transmit impulse, are strengthened by one class of intermediate filaments. Thus, the various kinds of intermediate filaments may function as a framework of the entire cytoskeleton. Okay, thanks for listening.